Hello, and welcome back to HSC Silicon Valley Youth Organization's biology lessons. My name is Minji Kim, and today we will be covering chemical reactions and enzymes. So, first, what exactly are chemical reactions? Well, they're basically where you get one set of chemicals and then they change into another set of chemicals. For example, a very common example would be water and oxygen going in and then carbon dioxide coming out, where mammals breathe air and release carbon dioxide. So the reactants are basically the input and the product are basically the output. Reactants in this case would be oxygen and water, and product is CO2 or carbon dioxide. Some chemical reactions are slow, like rust. They take time to happen, while others are fast and instantaneous, for example, explosions. Chemical reactions always involve the breaking of bonds in reactants and forming new bonds in products. So the bonds in the oxygen and water break and they reform to make carbon dioxide. In chemical reactions, energy is always either released or absorbed. Chemical reactions that release energy are called exothermic reactions and they occur spontaneously. Chemical reactions that absorb energy, endothermic, won't occur without a source of energy. So, like an outside source of energy that keeps feeding it energy for it to work. Here we can see an exothermic reaction, and we know that it's an exothermic reaction because it released energy, so there's less energy at the very end of the reaction than at the very beginning. Here we can see the activation energy, or the amount of energy that it needed to start this chemical reaction. Right here. That's the amount of energy needed, and that's the activation energy. Here we can see an endothermic reaction, and we know that it's an endothermic reaction because there's more energy at the end of the reaction than there was at the beginning. And here you can again see the activation energy needed. Now that we've gone over the basics of chemical reactions, we can move on to enzymes. What exactly are enzymes, you may ask? Enzymes are proteins, and they are the biological catalysts. And so this is why this actually goes into the biology section instead of the chemistry section, even though it's more of an overlap. Enzymes are biological catalysts and they generally end in ase, like catalase or ligase. They work by lowering the activation energy needed for a chemical reaction. They also speed up the reaction rate of the reaction. Here are some characteristics of enzymes. They start chemical reactions by lowering the activation energy needed. Here you can see that happening where the uncatalyzed reaction, or in this case it's talking about catalase, which is an enzyme found in your body and it breaks down hydrogen peroxide. And here you can see that the activation energy when there was no enzymes is quite large and there's a lot of energy needed. But when they introduced the enzymes, the amount of energy needed significantly decreased, and it looks like it's about half the energy needed. Enzymes also control the rate of the reaction, so it can make it faster or slower depending. But it's generally faster. Enzymes are also not used up after one use, so they're reusable, and they're more effective in smaller amounts, so a couple enzymes in a large amount of substrate. When you want to learn about how the enzyme actually works, you need to know three things. Substrate. The substrate is the material that goes into an enzyme. The active site is the place that the substrate binds to or attaches to on the enzyme. The product is the thing that comes out of the chemical reaction, which is the products. So here you can see the substrate, and along here is the active site, and the gray thing is the enzyme. you can see that the substrate attaches to the active site, and the active site actually, you can see, changes a little. The shape becomes a little different as the substrate binds to the active site. Then something happens, so a reaction happens, and we can see that the bonds were broken in the substrate, and out comes two products. Enzyme action. Enzymes are highly specialized, so they only work with one kind of substrate, and they do only one thing. For example, catalase only reacts with hydrogen peroxide, and that's the only thing that it reacts to. They are like a lock and key where there's one enzyme for one substrate. So we need that specific key to open that specific lock, similar to the enzyme and the substrate. So the induced fit model is basically where the enzyme actually molds around the substrate in order for it to fit. So you can think of it as like a catcher's mitt, where when you have your hand limp in the mitt, the baseball will not stay in there very well. But if you wrap your hand around the baseball, then it stays in place, and it like fits. So here you can see that this shape over here doesn't actually fit the substrate very well, but as the substrate comes in, the actual shape of the active site changes. So there are a couple conditions that need to be met in order for an enzyme to actually function properly. The temperature needs to be optimal, and the pH levels also need to be optimal. And if they are not perfect, then the enzyme will start to denature and no longer function. So when we say denaturing, it's basically where the shape of the enzyme changes. So the substrate no longer can bind with the active site anymore. And it changes so much that even when you use the induced fit model where it changes a little, it still doesn't fit anymore. So here you can see that a temperature on enzyme, um, and we can see that they work optimally from about 35 degrees to 45 degrees Celsius. And after 45 degrees Celsius, we can see that the enzymes start to denature and no longer function properly. Some other factors that can affect enzyme function are concentrations of the substrate or the concentration of the enzyme. As we've mentioned, enzymes work well when there are a few select amounts of enzymes in a large amount of substrate. Another two things that can alter enzymes' functionality is inhibitors and activators, and they work with how well the enzymes work when these things are introduced. That's it for this week's video, and I hope you enjoyed and learned a couple things. Thank you!